Hey everybody, welcome to the next lecture for human anatomy. This should be the last chapter that we do before we get into the real anatomy stuff. This should be the last kind of biology related chapter that we do. So let's get into it. So we want to end this kind of bio review with talking about DNA and how the nucleus works and some processes that the nucleus does. So in order to talk about the nucleus and the hereditary material that's in the nucleus, we have to talk about DNA. DNA the term DNA stands for deoxyribonucleic acid. If we break this down, D means to remove, oxy refers to oxygen, ribo refers to ribose, and nucleic acid, refers to a type of macromolecule that we spoke about a couple of lectures ago. I'm going to talk to you more about this when I show you the actual molecule. But basically what you're doing is you're removing an oxygen from ribose to get deoxyribose. And then you're adding that to a nucleic acid, which gives you deoxyribonucleic acid. It's a long thread-like molecule, varies in length between organism. 46 DNA molecules or chromosomes, is what is found in human cells. Human cells have 46 chromosomes. Other organisms have more. Some organisms have less. Bacteria only has one chromosome. Starfish might have a different number. Rabbits might have a different number. Algae, fungus, they all have different numbers of chromosomes. That's kind of what makes organisms unique. It's the amount of chromosomes they have, plus the arrangement of the letters in those chromosomes. The average human DNA molecule is about 2 inches long. And DNA and other nucleic acids are polymers of what we call nucleotides. So we had mentioned that proteins are polymers made of amino acid monomers. We mentioned that carbohydrates are polymers made of simple sugar monomers. DNA and RNA, which is the other nucleic acid that we're going to talk about, RNA, are polymers made up of monomers called nucleotides. A nucleotide consists of these three things: a sugar, a phosphate group, and a nitrogen base. And that's what we're going to get into now. This is the structural formula for DNA. So like we just said, a nucleotide is a sugar. And the sugar that we're going to be using for DNA is called deoxyribose. Anytime you see OSE, I might have mentioned that on a previous lecture. OSE means sugar, like glucose, fructose, dextrose. Ribose itself is a sugar. It's a 5-carbon sugar. And regular ribose, when you draw it out, at the bottom it has 2 OHs, like that. That's what ribose looks like. Deoxyribose, I'm not going to draw it out, but deoxyribose is the exact same thing, but instead it only has 1 OH. We have taken one of the O's away from the ribose, and now we call it deoxyribose because it's missing an oxygen. So that's the only difference between ribose and deoxyribose. That's the difference between DNA, deoxyribonucleic acid, and RNA, which is ribonucleic acid. It's regular ribose instead of deoxyed ribose. There's going to be a phosphate group attached in this nucleotide, which I'll show you in a moment in a picture that's going to be drawn way better than my drawing. And there's going to be a nitrogen base. What are nitrogen bases? In DNA, the nitrogen bases are called A, T, C, and G, and they stand for these words right here: adenine, guanine, cytosine, and thymine. Now there's a last one down here called uracil, but uracil is only found in RNA. So that's really important that we know that. That's why it says DNA bases are A, T, G, and C. These are DNA. For RNA, it's the thing with RNA is you replace T with U. So let me just erase this 1 second so I can write again. U replaces T in RNA. So you are not allowed to draw, when talking about RNA, which we'll talk about what that is in a moment, when you're drawing out RNA, you cannot put any T's in RNA. You have to replace them with U. So anywhere you would put a T, you're going to put a U. But if you're talking about DNA, it's A's, T's, G's, and C's. Okay. This is a nucleotide. Okay. So here is my deoxyribose, and you can see there's my missing O. If this was ribose, if this was RNA, it would be the exact same structure, except there would be another O next to this H. Okay, that's the only difference. Actually, the O would probably be on this side, but that doesn't matter. Okay, but this is deoxyribose. This is deoxyribose. So we're talking about, this is a DNA nucleotide. Okay, so here's our sugar. That's one component of this nucleotide. We have our phosphate, which is our second component of our nucleotide, and that's nothing more than a phosphate molecule surrounded by oxygens. And then we have our nitrogen base. Okay, our nucleic acid. Okay, this is adenine. This could have been thymine. This could have been guanine. This could have been cytosine. Okay, this could not have been uracil because it's deoxyribose, which means this is DNA, and there's no U's in DNA. There's only A's, T's, G's, and C's. So this example has adenine, but it could have very easily been thymine, guanine, or cytosine. It doesn't matter. All the nucleotides have the same structure, except this would change slightly depending on if it was one of these four. Okay, if this was ribose and not deoxyribose, and there was another O here, okay, this could be adenine, guanine, cytosine, or uracil. Okay, if that was just regular ribose, but it's not, and because it's not, it's deoxyribose, which means this is DNA, which means this can only be A, T, G, or C. Okay, but that is the structure of a nucleotide. These nucleotides get combined to form the DNA helix, or the DNA helix that you might know about. Okay, but you have to put all these together. Okay, you take millions and millions and millions of these, and you put them together, and they form the helix of DNA. Okay, so here are all the nitrogen bases. Okay, adenine and guanine have a special shape, and you can see that they are double ringed. All right, so this is one ring, and this is another ring. Here's one ring, here's another ring. There are slight chemical differences, right? We see, a double bonded O in guanine that we don't see in adenine. Okay, there's differences in each. That we have a hydrogen here, missing here that we have here. Okay, so there are differences, but the overall shape of adenine and guanine are the same. So because they have the same shape, we call them purines. These are the purines because they're double ringed structures. And then the other ones, cytosine, thymine, and uracil, they're only single ring structures. And obviously we see differences, right? We see differences in these things all over the place, okay? But they have the same basic ring structure. But instead of calling them purines, because they have two rings, we don't call them that. We call them pyrimidines. Okay, so we need to know that adenine and guanine, A and G, are purines, and that the C, T, and U are pyrimidines because they have single ring structures instead of double ring structures. And these are our options to make nucleotides, right? This structure could be attached to a phosphate and a ribose, or this structure, or this structure, or this structure, or this structure. Okay, depending on if we're talking about DNA or RNA. Okay, this is only found in RNA, where these are found in DNA. Okay. Okay, so DNA has this famous double helix shape, all right? This really shape that is kind of known by everybody nowadays. It's only been, it was only discovered in like 1953, 1955, the actual structure of DNA by a group of guys named Watson and Crick. Okay, but it resembles a spiral staircase, and they were the scientists that figured out how to what the shape was. A lot of people no one understood what the shape of DNA was. They knew it was important, but they didn't know the shape, and they figured it out. Okay, and I'm going to show you a picture that explains these two sentences. It says each side piece has a backbone of phosphate and ribose, deoxyribose. Okay, we call this the sugar dash phosphate backbone. And the sugar phosphate backbone is strictly structural. There's nothing about the sugar phosphate backbone that differs between organisms. The sugar phosphate backbone is the same in humans as it is in bacteria or fungus or plants. The difference that DNA makes or creates in organisms is found not in the backbone, but in the center of it, which I'll show you in a moment. Okay. So here's a picture of DNA, two pictures of DNA. This is like a ball and stick model that shows you the atoms in 3 dimensions with, with spheres, right? And all these different colored spheres are different atoms. Okay. And it shows you the spiraling nature here, right? It would just keep spiraling around and around. This is what DNA looks like if you were to draw like the molecular formulas, right? Kind of, or the backbone, the skeleton of a molecular formula. So what we see here, and you have to be able to identify these things on an exam. Okay, these circles, these are the phosphates. All of these little yellow circles are the phosphates. Okay, and all of these green pentagons are my ribose sugars. I'm going to draw an S for sugar. Okay, so all these little pentagons are sugars, and that's what forms the sugar phosphate backbone. Okay, sugar phosphate, get it? Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate on both sides. So think of it as a ladder, right? Here's our ladder, right? The outsides, the legs of the ladder, or whatever you want to call them, those are the structural components of the ladder. If you take these away, the middle parts have nothing to hold on to, right? And then in the middle of DNA, in the very center of this sugar phosphate backbone, are our nitrogen bases, A and T, G and C, T and A, G and C, okay? And that is where the genetic information is going to lie, okay? Not that any other organism has different letters, because all organisms share A's, T's, G's, and C's, right? If you're a bacteria or if a fungus or any other animal, all organisms are going to share these same components. All organisms have these phosphates, these deoxyriboses, these A's, T's, G's, and C's. It's just how many of these A's, T's, G's, and C's do we have, and what's the order of those A's, T's, G's, and C's. That's what makes you different than any other organism, or even the person next to you, for that matter. It's the amount of these letters and the order in which those letters are placed. Okay. You see little dashed lines, and I'm going to mention this in the text, but these little dashed lines are called hydrogen bonds. And we learned about hydrogen bonds in a previous lecture, and those are very weak bonds, right? They're not there's no actual any interaction between electrons, between the atoms, right? So this G is not sharing or giving up any electrons to C, but it has a little magnetic attraction to it because of their charges, right? So we call them hydrogen bonds. Very easy to break, right? We can break these bonds very easy. All you have to do to DNA to break it apart is to heat it up, okay? It's not very easy to heat up DNA when it's inside of cells, but if you had DNA in a test tube, it would be very easy to break it apart. Okay, so again, nitrogen bases, these are my A's, T's, G's, and C's. They form pairs, right? They form pairings. And if we go back, this is a pairing. This is called a base pair, right? Each one of these is a nitrogen base. A is a nitrogen base, T, G, C, these are all nitrogen bases. When they bond together here through this hydrogen bond, we call that a base pair, right? So in this picture, we see 4 base pairs, or actually 5. This is 1, 2, 3, 4, 5 base pairs, okay? Now what you'll notice is that certain letters like to bond to other letters. We always see that A always bonds to T, and we see that G always bonds to C. It doesn't matter what side the G's or A's or T's are on, but we always see that A and T bond and G and C bond. Okay, that's going to be very important. Okay, so back to this. A purine on one strand will always bond to a pyrimidine on the other, right? So A, which was my purine, is always going to bond to T, which is a pyrimidine. C, which is a pyrimidine, will always bond to G, which is the purine. Okay, so these are my base pairs. A's will always bond to T's, C's will always bond to G's. This was discovered by a person named Chargaff back in the 50s, might have been the 40s, late 40s, early 50s. Chargaff, okay, that was the name of the scientist. And he found out that A's always bond to T's and G's always bond to C's. And it is now termed Chargaff's rule. Okay, I don't call it the law of complementary base pairing because I want to give props to Chargaff. We call it Chargaff's rule. Okay, and Chargaff's rule says that A's always bond to T, C always bonds to G. Okay, the way he found that out is pretty interesting. Every time he studied the DNA of organisms or the genetic material of these organisms, he always found that the percentage of A always equaled the percentage of T, and that the percentage of C always equaled the percentage of G. So he figured out that there must be some type of relationship between A and T and C and G if they always show up in the same percentages. And what they discovered was that there's a bond between them. Every time you find an A, you find a T. That's why the percentages are always the same. So on an exam, if I tell you that an organism's genome is 15% A, you could tell me what the percent of G is. How? Like this. If A is 15%, that means that T must be 15%. A and T equals 30% of the entire genome, right? If you add those up, it gives us 30%. What's the remainder, right? You have 100% of a genome minus the 30 that we know about. That equals 70% remaining. So if I half of this must be G, half of this must be C. So 35% must be G, and 35% must be C. So the answer here is 35%. Okay. You might want to pause and rewind that and listen to it again just in case you didn't get it. Okay. Is there anything else I want to learn? No. Okay. Okay, so now we look at the term gene. Okay, a gene is a segment of DNA coding for the synthesis of specific proteins. Okay, and the genome is all the genes in a person or in an organism. Okay, we're talking human anatomy, so we're going to talk humans. But a gene is a portion of your DNA, right? So we have all this DNA inside of us. And a portion of the DNA is called a gene. And this portion is responsible for synthesizing or creating what we call a protein. And that protein can be structural, that protein can be an enzyme, that protein can be muscle tissue, that protein can be a communication protein between cells, that could be a receptor protein, it could be a channel protein, doesn't matter what it is. Okay, but that gene is responsible for that protein, and that protein is going to give you some type of trait. Right? Now, the trait could be blue eyes, or the trait could be, blonde hair. The trait could also be that you have certain markers on top of your cells. The trait could be that you metabolize things differently. So it's not always a physical trait that you see. It could be chemical traits or something like that. Okay. So the genome, like the Human Genome Project, we were trying to study where all the genes are in a human piece of DNA. That's what the Human Genome Project was. Okay, it took 10 years. 2 companies tried to race to get it done, and they both did it. And we know where the genes for diseases are. We know where the genes for just about every trait that we know about, we know where they are in the genome. Okay, we know about right now that we have about 20,000 genes. We thought we had millions of genes. Before the Human Genome Project was done, scientists believed that there were millions of genes. We found out as we discovered more things that we were way off, and there's only about 20,000 genes. Still a lot of genes, but a lot less than what we thought. Okay, only about 2% of your DNA is actually genes. Okay, only 2% of your DNA. And that's why we thought there were so many, right? So if you have, let's say that's all of our DNA, right? We saw this and said, wow, this big piece of DNA must hold millions of genes. And then we found out that 98% of all the DNA we have, right? So this much, 98% is called non-coding DNA, which means it's not responsible for making proteins, and therefore is not responsible for giving you traits. Only 2% of it is responsible for giving you traits. So instead of us thinking we had millions, it ended up only being 20,000. Now, it's not all at the end like that. Okay. If that was our piece of DNA, maybe this piece and 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 this piece are coding, and all the other stuff is junk. Okay. Okay. But only 2% of your entire genome is coding. The other 98% is not coding. Okay. But it does play a role in chromosome structure. You might be saying, well, what's the point of it? Okay. It plays a role in the structure of your chromosomes. It also helps regulate the activity of the genes. We don't want genes on or off all the time. And we can talk about that a little bit later, regulating genes. Okay, now I want to talk to you very quickly about the idea of chromatin versus chromosomes. Okay, these two words refer to the same thing, but in different forms. That's all that means. Okay, so very simply, a chromosome is a very tightly supercoiled piece of DNA. Okay, now I like to do this in class with a rubber band. I don't think I can do it here. But if you took a rubber band and cut it so that it's just one long piece instead of a circle, and you held one end tight and you twisted the other end and twisted and twisted and twisted and twisted and twisted and twisted. Okay, eventually what would happen is that the rubber band would start to, if you took the pressure off the rubber band and put your fingers closer together, the rubber band would clump up into this tiny little coagulation of rubber band. Okay, because of how much it's turning and twisting, it's getting smaller and smaller and smaller. As you turn it and twist it, it gets tighter and tighter and tighter, and the rubber band gets tinier and tinier and tinier. That's what a chromosome is. Okay, a chromosome is nothing more than DNA that's been twisted and 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 twisted until it forms this small twisted clump instead of this long, big, straight piece. Okay. Chromatin is the same exact thing. However, it's uncoiled DNA. So think of it more or less like a plate full of rubber bands, right? If we're going to use the same rubber band analogy, or a plate of spaghetti if you want, right? If you had a plate and you had spaghetti on it, the spaghetti would be going in all different directions, right? It's just loose spaghetti strings, okay? Whereas if you supercoiled all these spaghetti strings, they would look like something like that. Okay, it would take up a lot less space on the plate if you coiled them all up. And that's why we do that. That's why your body has chromosomes sometimes in your cell, and sometimes your body takes those chromosomes and uncoils them and makes them chromatin, right? It all depends on when your body and when your cells are doing certain things. When you want to go through mitosis and you want to divide your cell into two new offspring, we make chromosomes. That's when we see chromosomes. They'll look like these little X's. Okay. And we take these chromosomes and we make them into, we take our DNA and we make them into chromosomes so that we can shuffle them up and divvy them up equally into new cells. When we're not going through mitosis, and that's like 90% of the time, this is a very short amount of time that we go through mitosis. The rest of the time, we're just living our life cycle as a cell. We don't want our DNA to be condensed and supercoiled. We want to be able to read it because that's what DNA is. DNA is like an instruction manual. And you can't read a manual if the book's closed, right? So during the rest of our life cycle, this DNA is open and free, okay, like the plate of spaghetti, so that we can see the information inside and reference that information and look at the instructions, okay? So that's the only difference between chromosomes and chromatin. It's the same thing, right? It's DNA. It's either uncoiled as chromatin or it's supercoiled in like chromosomes. It's the same thing. Okay, very easy. Okay, here we have some diagrams to kind of show that to you. So here's uncoiled DNA. Okay, and as we coil it up and coil it up and coil it up and coil it up, it forms this thing. Whoops, I'm going to do it again. It forms this thing called a chromosome. So here we have a chromosome, and this whole area is basically chromatin. It's uncoiled or not supercoiled, and this is super, 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 super coiled DNA. Okay, so a minute ago I told you that a gene is an area of DNA that codes for a protein. So that was the definition of old. And I don't disagree with the new definition, but I don't see why it's so important to change it, but I get it, so I'm going to tell it to you. So, I've been teaching for 18 years now, And this was always my definition of a gene, a segment of DNA that carries the code for a protein. That is not false. Okay, that's not false. That is a true statement. But we've altered it a little bit to have this definition. A gene is an information-containing segment of DNA that codes for a piece of RNA that plays a role in making a protein. It's basically saying the same thing, but it's just adding something in the middle, right? So the way, and I'm going to teach this to you in a moment, you go from gene to RNA to amino acid to protein. That is the sequence that we all have known about for a long time. So we've always defined a gene as the piece of DNA that makes proteins. We were just kind of lazy in mentioning this. We knew this happened, okay, all the time, but we just never mentioned it in the definition. We just said a gene is something that codes for a protein, which is true, because at the end you get a protein. But now they want to say that a gene is a piece of DNA that codes for RNA that plays a role in synthesizing proteins, right? And proteins are made of amino acids. Okay, there's your amino acid sequence. Okay, that's the only difference. But you do want to know that for the exam. Okay, so what is a gene continued? So humans have, like I don't know if I mentioned it, but humans have 46 chromosomes. Okay, 46 chromosomes come from are in every human being's cells. Well, I don't want to say that. 99.9% of human beings have 46 chromosomes. Sometimes you have an extra one, and that can result in something called trisomies, which could be like Down syndrome. But we're going to talk about that in a different chapter, especially when we get to reproductive systems. Okay, but 99.9% of human beings have 46 chromosomes. 23 of those came from mom, and the other 23 came from dad. Okay, or one parent and the other parent. The genome, your entire set of DNA, is all 23 pairs of chromosomes because they come in pairs, just like shoes come in pairs, right? So dad gives you a chromosome 1, mom gives you a chromosome 1. Dad gives you chromosome 2, mom gives you chromosome 2. 3, 3, 4, 4, 5, 5. This goes all the way down to 22. And then there's something called sex chromosomes, okay? So moms will give you X's all the time. Dads will give you an X or a Y. Okay, sometimes they give you both, but again, we're talking about 99% of the population that is not born with abnormalities. So it's either going to be an X or a Y. All right. So if mom gives an X and dad gives an X, this baby is going to be a female. If mom gives an X and dad gives a Y, this organism will be a male. Okay, but they come in pairs. This chromosome 1 shares information with this chromosome 1. Okay, might not always be the same information, right? This chromosome number 1 might deal with eye color. This chromosome number 1 might say blue. This chromosome number 1 might say brown. So the versions of these chromosomes might be different, but the general information is the same. And that's why we come in pairs. Okay, that's why they come in pairs. Okay, you have about 3 billion nucleotide pairs in the human genome. So those A's, T's, G's, and C's. You have 3.1 billion of these pairs, which means how many letters do you have, right? If you have 3 billion pairs, that means if you're just counting letters instead of pairs, you have 6.2 billion letters, right? You have 6.2 nucleotides. All right, 6.2 billion nucleotides. 3.1 pairs, 6.2 total. Okay. The idea of genomics, or the subject of genomics, is the study of the entire human genome. Okay, how genes and non-coding DNA interact to affect the functioning of an organism. Okay, that's what genomics is. Very interesting subject. Okay, the genetic code. So nucleotides, like we said before, these A's, T's, G's, and C's, they code for an amino acid. Oops. Okay, like I said before, we have a gene. The gene gets turned into mRNA, which we'll talk about in a moment. That RNA is the code for an amino acid, and that amino acid is the code for a protein. or not the code, but it's the building blocks of the protein. Okay, so nucleotides, the gene, right, because the gene is made of A's, T's, G's, and C's, give code for the amino acids. This is the code. Okay, and then those amino acids get put together and you form a protein. The body can only make, now the whole thing lies in this amino acid section. Your body can only make 20 amino acids, okay? And these 20 amino acids are encoded by the genes made by A's, T's, C's, and G's, okay? So let's talk about how this works a little bit, okay? So there is a process. So now that we understand the structure of DNA, right? So we understand the structure of DNA is a double helix. We understand that the outsides are phosphates and sugars. We understand that the middle is going to be an A, T, G, or C, right? We understand that, I hope. The way our body uses this, okay, how does our body use this DNA? It uses it in a couple of ways, and we're going to talk about one right now. And one way that your body uses this, or your cell uses it, is called protein synthesis. This is the making of proteins. That's what synthesize means, making of proteins. Okay? There are two steps to make proteins. We have something called transcription, and we have something called translation. Okay? So I'm going to draw this one more time. It goes from gene to mRNA to amino acid to protein. Okay? I don't want to get too deep into this because I don't think it's necessary for this course. Okay? But this process of the gene being turned into mRNA, and what is mRNA? I should mention that. mRNA stands for messenger RNA. It's a piece of RNA that is a message. It's going to tell something else some information, okay? The gene can't be used as a gene, right? So we talked about ribosomes. These little ribosomes have to read messages to make a protein. And these little ribosomes read mRNA. That's what they read. Okay, these ribosomes can't go into the nucleus and look at the gene. A message has to come from the nucleus and be given to this ribosome. This ribosome can't go into the DNA, look at the DNA. So the DNA, that's what this is, right? The genes are held on DNA, makes a message and gives that message to the ribosome. Okay? That mRNA message is read by the ribosome, and the ribosome calls for amino acids. And every time it calls for an amino acid, it puts them together. This amino acid plus this amino acid plus this amino acid gives us a protein. Okay. This process here, from DNA to mRNA, this message creation, that's called transcription. Okay, the process of making this message from DNA to mRNA is the message making. That's the transcription. The process of taking that message and turning it into a protein and reading the message, this process, reading the message and turning it into a protein, that's called translation. Okay, so this message creation is transcription, DNA to mRNA, and turning that mRNA into a protein with the use of a ribosome is called translation. Okay, we've transcribed, we've written down. That's what transcribe means, right? We've made a copy of. If you wanted to, that's what monks used to do in the old days. Before they had printing presses, when they wanted to make a new Bible, they would transcribe a new Bible. They would read one sentence and rewrite it on another piece of paper. They would transcribe it. So that's what we're doing here. We're taking this information, we're making a copy, we're making a message out of it. That's transcribing. Then we have to translate that message into a protein. So just like the monk, they would translate the Bible into another language, right? So they would make the copy and then translate the copy. Okay, so they'd make kind of two copies, right? So here we have our info, we have our base information. We make a copy of that information called a messenger RNA. Then we take that message and we use the message to do what it tells us to do, which is make the protein or translate. Okay, hope that makes sense. Another process that we use DNA for is called DNA replication. Okay, when we want to make new cells, this is our parent cell. These are our offspring, right? This is called mitosis. Whenever we get damaged cells, when we have a cut or something, we want to make new cells to fill in the gap. When we want to grow and develop, we want to take cells that are already existing and make more of them. This parent and this offspring are genetically identical to one another, right? So if this was a skin cell, and this skin cell divided and made two new skin cells, we want these skin cells to be identical to this skin cell. We don't want these to be mutated in any way. We don't want them to be liver cells. We want them to stay skin cells. So we have to make exact copies of our cells. How do you make an exact copy? If this parent cell has 46 chromosomes, then these cells have to have 46 chromosomes. So how do we do that? How do we take one cell, split it in half, but they still both have the same amount of chromosomes? Shouldn't these have less? Shouldn't these have 23 chromosomes in them if the cell splits? No. Why? Because we replicate the DNA before we split. Okay? So if I have 46 and I didn't replicate and I just split it up, I would have 23 and 23. But because I'm not doing that, I'm taking 46 and then I replicate my DNA. I duplicate my DNA to 92 and then I split it in half. Here is my original cell. Here are my two new cells. I have two exact copies of the original. Okay. The process of that is called DNA replication. Okay. And before your cell divides, it goes through these 4 steps. It unwinds. Okay. So here's our wound up DNA. Right. It looks like a double helix. Right. It looks like this spun up staircase. It's wound up. It's tight. It looks like chromosomes. Right. First thing we have to do is unwind it. So during unwinding, we go from this to this. All right, we go from a twisted ladder to a straight ladder. We unwind it. And then we have to unzip it, right? We have to open it up. So instead of it looking like a ladder, it looks now like a zipper. Like that. Okay, we've unzipped it. So first we unwind it, then we unzip it, and then we build new strands. Okay, so let me grab a different color here. So then we build new strands. We take the zipper. Actually, okay, we take the zipper and then we start to copy it. And we make new strands. And what eventually happens is this. We get 2 new pieces of DNA. One which was the original, one side of which this new piece of DNA was the original, the blue one, and the red one was the copy. Here, same thing. The blue one was the original, the red is the copy. Okay? And once we have this, we can then repackage it and make it like the first one. Okay, we repackage it and then we're back to the beginning, but we have 2 of them. Right, we'll have 2 of these. And what we've done was we've replicated DNA. We started off with 1 piece just like this. We've unwound it, we unzipped it, we replicated it, and we repackaged it. And now we end up with 2. Okay, we had 1 and now we have 2. So we have successfully replicated our DNA. Okay, here are the steps, just in a little bit more detail so you can read those. There's an enzyme that helps do that. Okay, there are enzymes that are going to help to do this unpacking and unzipping and all this other stuff. Okay, so to unwind our DNA and to unzip our DNA, we have something called helicase. Anytime you see ASE, it's an enzyme. Helicase helps to unzip. Okay, and to build new ones, we have polymerase. Okay, helicase is the enzyme that unzips. Polymerase is the enzyme that makes new strands. And here's that polymerase word right there. Okay. Here is a diagram of what I just drew. So here is your DNA. Okay, it has to be unwound. Okay, here is helicase. The helicase is unwinding, unzipping it. We can see the zipper coming apart here. Okay, and here is our polymerase. This big green thing is polymerase. And this polymerase is copying the sections. It's making the new segments. And you can tell the new segment from the old because the old segment is blue, the new segment is gold. Okay. So the helicase unzips it, the polymerase makes copies. It sees an A, it makes a T. It sees a T, it makes an A. It sees a G, it makes a C. So it's reading half the DNA and then adding the other half as a copy. I know it's a lot, but this isn't a biology course, so I don't want to go too deep into it because this could be an entire unit, entire chapter just on DNA replication. Okay, the cell cycle. So that idea of mitosis, right? Okay, the idea of mitosis, this cell splitting up into two new cells. Okay, there's a cycle that makes this keep going and going, right? Because then eventually these cells are going to split and make cells, and these cells are going to split and make cells, and so on and so forth, right? We call this life cycle of a cell from when it's a single cell to when it divides. We call that the life cycle of the cell, okay? There are two major parts of the life cycle. There's interphase, and there's mitosis, and then there's a third portion, which I'm assuming is on the next slide, called cytokinesis. Okay, so that we can split this life cycle up into three parts: interphase, mitosis, and cytokinesis. Interphase, and I'm going to go through this here instead of going through it all on the slides. Interphase is like 95% of the cell's life. Okay, and we have three phases of interphase. There's the G1, the S, and the G2 phase. G stands for growth. S stands for synthesis. So in this first part of this cell's life, you go through this G phase. And then I think the next slide has this nice, yeah, it has this. So here's our cell. We're starting over here. Okay, that's going to be our starting point. Again, it's a cycle. There's no beginning or an end, but this is where we're going to start. Okay, so it's a new cell. It's just started its life. Okay, it's going to go through, the first thing it's going to do is go through this G1 phase where it grows, it gets bigger in size, right? Because if we take a cell and we split it in two, these two new cells are smaller than the original one, right? If you have a peanut butter sandwich and you split it in half, the two halves are smaller than the original. So this cell starts off smaller than its normal size, and it has to grow a little bit. And that's what G1 is going to happen. It's normal growth. It's going to get larger in size, larger in mass. Okay, and then it's going to hit this S phase. S stands for synthesis. And this is where our DNA goes from 46 to 92 chromosomes, right? We have to replicate our DNA here. This is where DNA replication happens. Okay? And we have to do it here because it's going to split soon. It's going to split over here. This is mitosis. That's where it's going to split. Okay? So we have to do it before that. So we do it over here in this S phase. And then we hit the G2 phase where we prepare for mitosis. We prepare to get the split happening. And what happens there, we find the production of centrioles, which you saw in the previous lecture. Okay, our nucleus dissolves. A lot of things are going to happen in order for the breakdown or the division of this cell in mitosis. So all this green area, this is all interphase. This whole area here is interphase. That's the first part of the life cycle. The second part of the life cycle goes from here to here, and that's called mitosis, okay, or the M phase. We can call it the M phase or mitosis, okay, or mitotic phase, same thing. And in mitosis, and we're not going to go through each one of these steps. Normally I would if this was a biology course, but we're not talking about biology. We're talking about anatomy. We don't need it. Okay. Each one of these steps is a different step in the splitting of this cell. And I'm going to have you memorize the order of these steps, but I don't want you to memorize anything that happens in these steps. Okay. I don't think that's important here. Okay. But the first step of mitosis is called prophase. The next step is metaphase. The next is anaphase. The next is telophase. And those are the 4 steps of mitosis. Okay. These steps lead to the division. And you can see here, we see some breakdown of the nucleus. We see our chromosomes lining up in the middle. Here we see our chromosomes being pulled apart. Here we can see 2 new cells almost completely disjoined from each other. And then once you get to the end of telophase, you hit cytokinesis. And this is my third portion of the life cycle. Cytokinesis, cyto means cytoplasm, and kinesis means movement. So this is the movement of cytoplasm so that you get two equal sized cells, right? If you had a peanut butter sandwich and you wanted to share with your friend, you would make sure that each side is an equal portion so that you don't, get anyone angry. Same thing here. We don't want any cells to be bigger than other cells, right? We want this cell and this cell to have an equal chance while they're developing. We don't want one to be bigger than the other or smaller than the other. We want them both to grow at the same rate and be the same size and everything like that. And then these two cells would then go start their own life cycle, right? This cell and this cell will go through G1. And then eventually they'll get to the S phase where they have to duplicate their chromosomes. And then they'll get to G2 where they have to prepare for mitosis. And then they'll get to mitosis where they split up. And then there'll be 4 cells. Okay, and all 4 of these will then start their life cycle. Okay, and that's how the cells go. Now, different cells will do this cycle faster than others. Skin cells do this extremely quickly. Nerve cells don't do this at all. Once you have a nerve cell, they don't divide. You can't get them back, right? So different cells, bone cells, this takes a long time to do because that's why it takes so long for bones to heal after a break. Okay, so some cells do this much quicker than others. Okay, and again, I don't need you to know the things in these different phases. I want you to know what G1, S, and G2 are, but I don't need you to know the difference in the mitotic phases. And I don't even have it here, which is good. Okay, I just want you to know the order, and it's P-mat. Just think of a dog, right? A dog, where's a dog go wee-wee? A dog goes wee-wee on a P-mat, right? A pee-pee pad, a wee-wee pad, P-mat. Okay, it's prophase, metaphase, anaphase, telophase. Okay, right there, P-mat, P-M-A-T. Why do we go through mitosis? Just to mention this. Okay, we want to replace cells that have been that have died. Okay, if you have cells that died, we want to replace those. If you have cells that are damaged due to injury, we want to replace those. And as you're getting larger and you grow, okay, you want to make sure you have more tissues. You want to make sure your organs are developed, and that's why we go through mitosis. Okay, we're not going through all these different phases. Okay, next up, heredity. Okay, heredity is the study of the transmission of genetic characteristics from parent to offspring. So your two parents, mom and dad, give you their DNA. You don't get a full set of mom's DNA. You don't get a full set of dad's DNA. You get a random mixture of mom's and dad's DNA. You might get 80% mom and 20% dad. You might get 15% dad and 85% mom. You might get 4% mom and 96% dad. It's completely random. That's why we don't have two people in the world that are exactly alike. Not talking about twins and things like that. But that's what heredity is. It's the transmission of genetic information from parent to offspring. A karyotype is something that scientists can do to make sure, or doctors can do to make sure that a person is developing properly and that they have the right set of chromosomes. We can do this in the womb. We can actually test to see what your karyotype is. So a karyotype is nothing more than a chart or a model that counts your chromosomes. Okay, you can take a picture of the DNA inside of fetal cells by taking them out through something called an amniocentesis, which is where you put a large needle into the womb of a mom and take out the liquid in the womb. That liquid has cells that came off of the baby, right? If you sat in the tub, your skin cells would fall off into the tub water. Same thing with the baby. The baby's sitting in the tub water, or the womb, in the liquid in the amniotic sac, and the cells are falling off that baby. So the idea is if you take out the liquid from the sac, you're taking out cells of the baby, and then you can look at those cells and count the chromosomes that are in the nuclei of those baby cells. And that's called a karyotype. And you're looking for 46 chromosomes. You don't want to see 47. You don't want to see 45 because that means there's some type of abnormality, which could be Down syndrome or some other diseases. Okay, you want to see 23 pairs that equal 46 total chromosomes. Okay, one chromosome from each pair is inherited from each parent. There are 20, and I said before, you have 22 what we call autosomal chromosomes. and you have one pair of sex chromosomes. The pair of sex chromosomes can either be XX or XY. Okay. The other 22 autosomal chromosomes are numbers 1 through 22. Okay, chromosome 1 through chromosome 22, one from your mom and one from your dad. Okay. This is a karyotype. This is a normal human karyotype. What we see a pair of 1s, a pair of 2s, pair of 3s, pair of 4s, pair of 5s, pair of 6s, all the way up to 22. We have pairs. Okay, we don't want to see a person missing a chromosome. We don't want to see a person having too many chromosomes, right? If there were 3 chromosomes here, that's called trisomy 21. Trisomy means you have 3. If you only have 1, that's a monosomy, okay? Both of which you do not want, right? You need to, in order to function properly, you need to have the proper amount of chromosomes. Here are, now, if this was a male, this box would not be present, right? And you would see one X chromosome and one Y chromosome. And if this was a female, this box would not be present, and you would see 2 X's. Now, again, you can have abnormalities of autosomal chromosomes, like trisomy 21, or you can have abnormalities of sex chromosomes. You can have 3 X's, called triple X syndrome. You can have XXY. Okay. There are lots of different combinations of, you can have XYY. Okay. But those are abnormalities. Those are not the norm. Okay. When talking about karyotypes, we have these 2 words called diploid and haploid. And we can even talk about that in mitosis, right? If we had mitosis, we have 46 chromosomes, which give you 46 and 46, right? That's called mitosis. We said this already. This 46 number is called the diploid number, right? Each parent gives you 23, but they're in pairs, right? So if I asked you how many, if you had 4 shoes, okay, you would have 2 pairs of shoes, but you would have 4 total shoes, right? So you have 46 total chromosomes, but you actually have 23 pairs of chromosomes. That 23 number is called the N number, okay? So as human beings, there are 23 unique chromosomes. But because they come in pairs, we multiply it by 2, right? Because each parent gives you a set. So mom gives you a set of 23, dad gives you a set of 23. So we multiply that by 2, and that gives us 46. We call that the diploid number. 46 is our diploid number. So all of our cells, except for our sex cells, and I'll talk about that in a minute, all of our cells in our body, except our sperm or ova, are diploid. So our skin cells, our heart cells, our brain cells, muscle, skeletal, endocrine, lymphatic, all of our cells are diploid, meaning they have pairs from both parents. Okay, they have 23 pairs from both parents, which gives a number of 46. So when a cell goes through mitosis, it creates diploid offspring. But then there's another process. So that was mitosis. There's another process called meiosis. Meiosis doesn't result in this. Meiosis takes a diploid cell and splits it into 4 cells with 23 each. These cells are called sex cells. If it was a male, they'd be all sperm. If it was a female, they'd be all ova. And these aren't called diploid because they don't have 46 chromosomes in them. They only have half the amount of chromosomes. They don't come in pairs. They're unpaired. Okay? So we have a different name for them. Instead of 2N, they're just N. And we call them haploid. So these cells, these gametes, these sex cells, we can call them anything we want. Not anything, but gametes, sex cells, sperm for male, ova for female. Okay, gamete, sex cell, sperm, and ova. These are all unpaired haploid cells. And the reason that they're haploid is so that when an egg with 23 chromosomes gets fertilized by a sperm with 23 chromosomes, they create a diploid cell, right? That equals a diploid cell with 46 chromosomes. And that new organism with 46 chromosomes who's diploid can then reproduce and make more diploid cells. Okay, the only cells in the human body that are haploid are these gametes / sex cells / sperm and ova. Okay, now we're getting into heredity a little bit more with the terms genotype and phenotype. Okay, what slide am I on? Okay, genotype and phenotype, these are things that we associate with genetics. Okay, if we wanted to talk about how a gene is going to get passed from one person to another, we have to talk about something called alleles. An allele is half of a gene. Okay, an allele, and this is me simplifying this greatly. Okay, half of a gene. Okay, so as I said before, so let's say we have a chromosome 1 and a chromosome 1, and they had to do with eye color. This is your mom's chromosome that she gave to you, and this is your dad's chromosome that he gave to you. A portion of this chromosome, I'm just going to circle it in blue or mark it in blue, a portion of this chromosome deals with eye color. Not the whole chromosome, just a portion of it. Okay? And your dad's chromosome says that your eye color should be green, and your mom's chromosome says your eye color should be brown. We'll just make believe that's brown because I don't have brown down here. Okay? This version of the gene that your mom gave you is one allele, and this version of this gene that your dad gave you is another allele. So you always have two alleles for a gene. That's why I call it half a gene. Okay, so this is one half of the gene, this is the other half of the gene. You put them together, they have to fight it out. Okay, and this is where dominance and recessiveness comes in. Okay, in real life, if you had someone with a brown-eyed allele and had someone with a blue-eyed allele, the brown-eyed allele would win because it's dominant. And we do that, we show that by using capital or lowercase letters. Okay, so for the brown-eyed allele, I would put a capital B. For the blue-eyed allele, I would put a lowercase b. Capital B is dominant, lowercase b is recessive, and whatever is dominant always wins, so this person will have brown eyes. Even though they have the gene, or half of a gene, they have an allele for blue eyes, they're not going to have blue eyes because this other allele that their mom gave them is dominant, which is going to result in them having brown eyes. Okay, so now what's phenotype and genotype? Okay, phenotype is an observable trait. So if you look at someone with brown eyes and you see that their eyes are brown, that is their phenotype, brown eyes. But you don't know what their genotype is. This is the genotype. Okay, that's the genotype. But this genotype could be different. This isn't, everyone's genotype for brown. Not everyone who has brown eyes has this genotype. What if there was another person and their mom and their dad, let's say mom gave them a brown-eyed allele and dad gave them a brown-eyed allele. This person would have capital B for brown, which results in, again, brown eyes. Therefore, their genotype is capital B, or brown, and this person's genotype would be capital B, lowercase b, or brown, blue. So that's the difference. Here, you're only looking at someone. Here, you look at something and you say, okay, that's brown. I don't know what version of brown it is. Is it this version of brown, or is it this version of brown? I don't know. It doesn't matter because that's what I'm just talking about, an observable trait. Okay, I just observe that this person's eyes are brown and this person's eyes are brown. So these two people have the same phenotype. However, these two people do not have the same genotype. This person's genotype is dominant, or capital B. This person's genotype is capital B, lowercase b. So their phenotypes are the same, but their genotypes are different. We can show this with something called a Punnett square. A Punnett square can take two individuals, and you have to know the parents. You take the parents' genotypes, and you put them onto the Punnett square, and the Punnett square can show the probability of organisms being born from those two individuals. So if I have an individual that is capital B, lowercase b for eyes, and another individual that's capital lowercase b for eyes, these are the possible combinations that I can get for their children, right? Because this is essentially a sperm, and these are essentially eggs, or vice versa. Okay, and if this sperm fertilized this egg, I would get brown eyes here. If this sperm fertilized this egg, I would get this type of brown eye, capital B, lowercase b. If this sperm fertilized this egg, I would get capital B, lowercase b, brown eyes. But look at this one. If this sperm fertilized this egg, they would get 2 lowercase b's. And that's how we get the blue gene. Okay, so this person, or these 2 individuals, these 2 parents, the chances of them having a child with blue eyes is 1 out of 4 boxes, right? 1 out of 4, or 25%. So these 2 individuals, if they have a baby, no matter if it's male or female, there's a 25% chance that that baby has blue eyes. There's a 75% chance that that baby has brown eyes. Because this would result in brown, this would result in brown, this would result in brown. That is the quickest I have ever taught simple genetics. But that's all we need for this course. Okay. Then we have sex-linked traits. I'm not going to get into Punnett squares with this, but all I want you to know is that some traits are carried on X or Y chromosomes. Okay. Most traits are held on the first 22 autosomal chromosomes. Most traits. There are some traits held on the X and the Y. Okay, most of the time, those X and Y chromosomes are going to hold traits that deal with your sex, right? They're going to deal with the production of breasts, reproductive organs, things like that, vocal cord thickness, things like that, different hormones like testosterone and estrogen. Okay, but there are some traits that are held on the X or the Y, and if they're held, and we call those traits sex-linked, okay, because they're linked to the sex chromosomes. Okay, one great example is colorblindness. Colorblindness falls on the X chromosome. This results in males having more of a chance of getting colorblindness than females, okay? Because if you have colorblindness for a female, you can have one chromosome that has the recessive allele for colorblindness, one allele that has the dominant trait for colorblindness, and this person is not colorblind because the dominant trait beats out the recessive trait, not colorblind. But if you were a male and this has the colorblind allele on it, this has the colorblind trait on it, the Y has nothing to do with colorblindness, only the X does. So there's nothing to fight this off. There's nothing to combat this, right? The only reason this person's not colorblind is because the big R beat up the little r. But here, there's no big R because the R's aren't held on the Y. The R's are only held on the X. So there's no big R to fight off the little r. Therefore, this person becomes colorblind. And that is a sex-linked trait that causes males to be colorblind more often than females. Same thing with hemophilia. Hemophilia is a trait where your blood can't clot properly. And that, again, is held on the X chromosome, so we can call it hemophilia H. Okay? And there's no H allele on the Y, therefore it cannot fight that recessive allele, so males have a tendency to be more likely to be hemophiliacs if their parents have it as well. So that is a very quick rundown of DNA structure, function, heredity, and genetics. And I'm sure you'll have lots of questions, but I told you everything you need to know. But if you have any questions, please put them in the comments section, or you can email me. And I'll see you on the next lecture.